Hello, everyone, and welcome to this month's IBEAM seminar, where we bring guest researchers from around the world to explore fascinating topics in chemistry. Now, before we start, I would like you to consider for a moment one of the most fundamental and enduring mysteries that sparks human curiosity: our origins. Now, the search into the origin of life has driven significant exploration, and at the heart of that lie the fields of prebiotic and systems chemistry, in which chemists try to understand how the building blocks of life come from non living matter, and how these components could have self organized into complex, self evolving, and self sustaining molecular systems. And there are individuals who dedicate their careers and focus their talents to answer these questions on our behalf. Motivated by a deep sense of wonder and genuine desire to understand our place in this universe, Professor Peter Strazewski is certainly one of these individuals. Now, for decades, Professor Strazewski has been guided by a powerful why: to understand the earliest critical steps in the chemical evolution that ultimately led to life as we know it. Now, his impactful work within his research group in Lyon, SysChem, seeks to carefully decipher and piece together how these biochemical systems could have assembled in the absence of enzymes or ribosomes, in addition to trying to pinpoint the possible role of Earth's geology. He deeply investigated interactions between nucleosides, oligonucleotides, and amino acids, seeking to shed light on the core components that might have initiated early peptide synthesis, a key area in developing biological systems, making significant contributions to the field. On a personal level, I feel honored to introduce Professor Strazewski, not only for his contributions, but also because he played a formative role in my own scientific development by teaching me in my master's. His passions and insights were truly inspiring and have left a lasting influence. Today, he continues to explore these fundamental questions, examining the fascinating interplay between different environments on early Earth through the lens of prebiotic plausibility. His presentation, How Nitrogen, Phosphorus, and Carbon Provided Atoms and Chemical Energy to Create the First Phosphate Esters, offers a new chemical perspective on how early Earth components could react to create the conditions for life, a question we all want answered. So please join me in extending a warm welcome to Professor Peter Strazewski. The floor is yours, Professor.
Thank you very much, Angelo, for this impressive introduction. I feel flattered. Hello, everybody in Umeå in the IBEAM department. I've never been to Umeå. I had to look it up on the map, and as a southerner, I tried to see, have I been so high up north? And maybe I was twice in Anchorage, which is on the other side, but in a similar biological and biochemical region. So the work that I will describe today is actually a summary of my last PhD student. She was collaborating between my group and Laboratoire de Géologie de Lyon, so geology. We don't have a department of biogeology, but geology. And there is an Institute of Origins, LEO, who actually funded her part of her PhD. And so she was a member of the geology lab, but her work she carried out in my group, in SysChem's group. So that's the Institute of Organic Chemistry. It's actually Institute of Organic and Biochemistry, Molecular and Supramolecular Chemistry and Biochemistry, ICBMS. And as you see, SysChem, so that's systems chemistry of biomolecules and prebiotic chemistry. My research group is part of a team which is called CO2, and CO2 is not carbon dioxide, but Chimie Organique 2. And so no wonder that since this is a collaboration between organic chemistry and geology, it's actually the first in Lyon. I'm a little bit ashamed about the fact that we started because it's late, we could have started much earlier. But no wonder that then there are several names in co-authors. So Anastasia Shvetsova is the long-term lab worker who did most of the experiments that I'll present you today. There was Augustin Lopez and Michele Fiore who supervised her. But since this is quite a complex project, there is also Linda Merzoud and Henri Germet. They are specialists for the density functional theory, so they just did calculations. And Elodie Fromentin and Anne Baudouin, they are specialists, or they are those who took dozens, several hundreds mass spectra and NMR spectra. Isabelle Daniel is a mineralogist. She is actually was the supervisor of this PhD, and I am the one who is very glad to be able to give you this talk. So as you see, the title that I chose, How Nitrogen, Phosphorus and Carbon Provided Atoms and Chemical Energy in the Late Hadean Era to create the first organic phosphate esters, is something which should be it should be seen in a scenario. And this scenario, well, I have to focus on the Hadean area. I have to focus on the first islands, because in the late Hadean area, we just had a huge global ocean and some few islands, we think. And so what we know is that at the time, the sun was dimmer. It was more UV active, but it was about 20 or 25% dimmer. The atmosphere contained mainly at the time already, mainly nitrogen, but had a lot of carbon dioxide, of course, water vapor, and in the vicinity of such islands, probably hydrogen sulfide coming out from the volcanic activity that was very intense in the area. Papers show that the temperatures in the crust and in the upper parts of the crust could still be about 2,000, 1,000 degrees, and then of course in the atmosphere everything evaporates and becomes cooler, and so there were the temperatures below 100 degrees, or else we would not have been able to have an ocean. And this means that that the condensation evaporation cycles of water was a normal thing. So all this part of the scenario, let's say, nothing special. However, there are other things that we have to consider. For example, the days were shorter, imagine, about 17 hours maybe, one day. So that has changed the rhythm, that is an accelerating effect for the chemistry that happened, because we need energy. The months were shorter. Let's not forget that we are talking about a time where about not much more than 100 million years after the Theia impact that formed the moon. So the moon was huge, very close, spinning around like crazy, and having its strong effects. The periods between ebb and flood were much shorter. The tides in general were much more intense than today. The winds were much stronger. The ocean waves were, somebody calculated, about 10-meter average. Today we have 4-meter average. So this means that the exchange processes that could have taken place on such islands were faster and more intense, and aerosols could bring in new matter that has been produced on land into the sea. So these thoughts are based on several later papers where we see that we cannot forget about the moon that was so close. Then, of course, there was a more or less intense impact bombardment, and for us in this work, what is important is that there was schreibersite, rocky metal meteorites that brought in nickel phosphide that when it fell on Earth into water or into wet areas, they would corrode to phosphites and phosphates. And if there was some sulfur around because of the volcanic activity, then also thiophosphates could form. Thiophosphates are not very, very stable, but there are conditions where many days thiophosphate can stay. So this corrosion produces hydrogen gas. This hydrogen gas leaves our planet. So the situation where we are thinking that we are trying to model as chemists, okay, organic chemists, is taking the fact that phosphorus containing minerals were on the Earth's surface. And for the time reasons, I told Angelo I had to cut this talk into two. So you just have to believe me for the moment that we think that there was a lot of cyanamide and urea also on the surface, and maybe also other non-volatile reduced nitrogen compounds. So what is important there is that the nitrogen, that is the most stable, N2, the most stable form of nitrogen, could be fixed into first simple compounds, which were cyanamide, urea, formamide, acetamide, and such molecules. So that's the scenario where we would like to model our experiments. What are the phosphorus sources that we used? So natural sources of phosphorus on the early Earth, as I said, phosphides, phosphite. These are reduced state phosphorus compounds. Orthophosphate is the most stable one. But on islands where the heat could go up quite high and the water would evaporate, pyrophosphate, maybe triphosphate. Some people think that cyclic trimetaphosphate could be around, at least a part of the time, in mixtures. When, as I said, when you have sulfur, then another form, milder form of activation in thiophosphate could play a role. So what is known, what I know from my dear colleague Isabelle, Danielle, is that chondrites contain inclusions that contain already phosphite, like whitlockite and biosite. And schreibosite, as the main source, would corrode into different other secondary minerals, and this also dependent on where they fall. So if there was cyanamide around or much sulfur around, then still other things would happen. So what we picked out as a interesting minerals would be vivianite and struvite. Vivianite as the ferrous phosphate, struvite as orthophosphate, but ammonium and magnesium. Then, of course, the most stable and unreactive and not useful for us chemists phosphate minerals are apatites, but they would weather in the around hydrothermal vents into, for example, canafite, which is a pyrophosphate mineral, and bifosamide, so dihydrogen orthophosphate with ammonium and potassium. So we chose these four source minerals, not literally, because we used vivianite, we used synthetic struvite, we used canafite analog, simply calcium pyrophosphate. And our phosphate sources, that compound that we used most was sodium dihydrogen phosphate. So with this short introduction, I'll step into our experiments. This is a carousel reactor, so you can make how many? 12 reactions at the time. You see that I'm not a lab worker anymore. 12 reactions at the time. You can heat them. We did heat them, or anesthesia, in open tubes. We stuck to the principle that we stay at one temperature, so mostly we used 115 degrees, sometimes or quite often 75 degrees, to see close to the evaporation temperature of water, a little bit below, a little bit above. And then sometimes with very unstable compounds only 65, and with very stable compounds we tried even 130 degrees, and we would then heat them for a long time, and no added water, only in control experiments to see the effect of water. So these were actually solid phase reactions or mixed solid liquid phase, because the compounds that were inside would then, one of the compounds would maybe be liquid, but not always. The scales that we worked at, the smallest scale was 0.5 millimoles, so several hundred milligrams. So we would heat it up to 5 days. Sometimes we made reactions in a large scale, not in the carousel reactions. This would go up to several grams. So these are reactions that are, you can see and feel and weigh and everything, not in Eppendorf's. It was important for me because before that, I did Eppendorf reactions with Michele Fiore, whom I will present you at the end. So the concept was the following. We had 3 kinds of compounds maximum at once. We had something that we can name a condensing agent. It could be too unreactive, then we call it a liquidizer, something that mobilizes solid stuff in these tubes. That's one component. The other is the phosphate source, and the third is the alcohol. So here is a table out of a paper that we have submitted, and the details are all in there. On the left column, you see the list of dehydrating or condensing agents. So dehydrating means a molecule that can hydrolyze into something more stable. In the first part, you see cyanamide. Cyanamide is a solid, but it melts at 44 degrees and it stays melted up to high temperatures. Urea, of course, is something which is quite stable, much less reactive than cyanamide, or the first hydrolysis product of cyanamide, and you can heat urea up to about 133 degrees, and then it will start decompose and produce ammonium cyanate, which is the reverse of this famous Wöhler reaction, and everybody knows that phosphate, inorganic orthophosphate, catalyze this decomposition. So the most stable part, the end products are ammonium carbonate or CO2 and ammonia finally. But we also used sometimes instead or in addition formamides and acetamides that in the literature people think that might have been around, maybe not much, but still. Acetamide and methylacetamide, formamide and methylformamide, these are all liquids at the temperatures that we worked with. So these we call liquidizers, also maybe catalysts, proposed as catalysts, thinkable catalysts. The phosphate sources, as I said, inorganic phosphate, you will, throughout the talk this will be PI, sometimes we'll see O18 labeled phosphate, and struvite, vivianite, thiophosphate as the commercial product is a tribasic trisodium salt. Here you see the canaphite analog, so it's a highly basic calcium pyrophosphate and tribasic cyclic trimetaphosphate. And then the alcohols. So we chose alcohols, and of course we chose also nucleosides, but we chose because I'm strongly linked to research in membrane forming molecules. We chose glycerol, we chose monopalmitoyl glycerol, that's MPG 6, bis tetradecanoyl glycerol 7, dioleoyl glycerol 8, dodecanol, ordinary dodecanol, then geraniol as a bit an unusual but still interesting or potentially interesting alcohol, then all the ribonucleosides, including 1-deoxyribonucleosides, thymidine, and just because, yeah, who knows, yeah, pyruvic acid is not an alcohol, but it has in equilibrium, it can produce an enol. So what I pick out for you here is the thermodynamic aspect of this table. In cyanamide, we have the strongest free energy that packed into the molecule that can be released and used to complexify other molecules. Sorry that it's not a kilojoule. I'm a kilocalorie person, and we have 29 about kilocalories per mole. CTMP is almost 10 kilocalories per mole in breaking the first bond. And then pyrophosphate gives you 4.5 kilocalories. So together we would have something like 15 kilocalories to use up in from CTMP to orthophosphate. Then urea is much less reactive, much more stable, but still is an important condensation agent. It delivers 3.5 kilocalories per mole and free energy to hydrolyze to or react to ammonium carbonate. And these values should be compared with, table values. Glyceryl phosphate delivers 2.2 kilocalories per mole upon hydrolysis. Normal other sugar and nucleoside monophosphates between 3 and 5 kilocalories. The condensed organic triphosphase minus 7 and to minus 12 kilocalories, and the highest free energy that you get from the hydrolysis of pyruvate, that's our least stable, most difficult to phosphorylate compounds. So why I'm showing this is, of course, in water this is not makeable, any of it. The thermodynamics is against that. So that was the main reason why we need to dry down the mixtures and to see what happens when there is no water around. So our analysis, just so you see before I show you the results, we use a lot of NMR. Of course, we use phosphorus NMR. So the first thing is, for example, this is glycerol, urea, sodium dihydrogen phosphate, heated for 5 days at 115 degrees, and then you extract it into DMSO, and you measure a proton decoupled phosphorus NMR. That's how it looks like, a typical mixture. So there is a zone, the least shifted zone around inorganic orthophosphate, which shows all acyclic phosphates. There is a zone downfield which shows 5-membered ring cyclic phosphates, and there is a upfield region which shows 6-membered ring cyclic phosphates. Now, when we stop the proton decoupling, we get couplings, and so, for example, in the middle of the slide, you see a triplet and a doublet. That's because the glyceryl phosphate is phosphorylated on the primary and the other one on the secondary alcohol. You see to the left the molecule that gives a doublet of doublets. To the right, you see a molecule gives a triplet of doublets, and it's absolutely clear. Many molecules, mainly the major ones, it's absolutely clear what they were in such mixtures. So we could, through looking exactly at the difference between the proton coupled and the proton decoupled spectra, we could find the residual PI, the residual the starting material that has not reacted, or, as you see here to the right, inorganic pyrophosphate. And then we would quantify the NMR, the phosphorus NMR signals through integration and take out all inorganic phosphorus signals, and then we call that consumption, PI consumption, or conversion. We don't call it yield because we never take it out, yes. But that's how we define our consumption, our conversion values that you will see later in the talk. So we also use proton NMR, of course, because many molecules contain other things than just phosphate. We have organic carbonated products here in the proton NMR very clearly. Many times we used HMBC, proton phosphorus HMBC, to unmess, to see more in a complicated mixture. But then the 1D spectra would be the ones that deliver the quantities. And sometimes we use, actually, we're happy to use a probe, an NMR probe that doesn't exist in Lyon, maybe now, but that was 3 years ago. So we went to Toulouse, Anastasia went to Toulouse. They have a head where they can decouple on protons, and you can make an HMBC correlation between phosphorus and carbon. This was very valuable for what you see in B, these two molecules. We would never have found out what these were, because you see, this is the acyclic area on phosphorus. Usually phosphodiesters are more upfield to the monoesters, as we will see, for example, in in other molecules. However, we had here two diastereomers and three compounds that produce downfield shifts, and they are still phosphodiesters. The simple reason is because they were carbonylated. They have this carbonyl group stemming from urea, of course. We would never have found out that. It was a beautiful way of, describing certain the development in time of certain mixtures. So when you follow here the arrows, the orange and blue arrows, you see exactly these compounds, how they start to be produced after 8 hours, and this is a time-dependent proton decoupled phosphorus NMR spectra of samples that were heated only for a certain time and then extracted. Yes. So when you do the same time dependence on proton NMR, you see when the carbonyl compounds develop, what happens with the urea, how the spectrum gets more messy and messy. And we used also C13 NMR. So here I add proton decoupled, of course, proton decoupled C13 NMR. Why did we do that? You see here a zoom in the carbonyl region of glycerol urea, N15 urea, sodium dihydrogen phosphate mixtures heated for 115 at 115 degrees for 5 days, and you get this. So what we did, we started with urea, N15 urea 1:1 with respect to glycerol and sodium phosphate, and what we wanted to see is whether we can prove through this reaction the presence of isocyanic acid, HNCO, or isocyanate. We could never directly see cyanate in these solid mixtures, but when we can observe singly labeled urea, then this would be a proof for the fact that isocyanic acid was present in the mixture. So the first, to the left, you see heating N15 urea mixed with natural abundance urea gives a singlet for the normal urea and a triplet for the N15, twice the urea that contains two N15s. You see an isotope effect, so they don't superimpose exactly, and after heating for hours at 100 degrees, nothing happens in DMSO. However, when we do the same heating in the presence of inorganic phosphate and glycerol, what you see here in the time in this stack is that first thing is urea degrades, of course, because that's what it does when you heat urea. But there is a doublet coming up, and this doublet, of course, is the appearance of the nitrogen exchange urea, all in C13. So we use these data to determine kinetic rate constants. So what you see in the zoom is, let's look at the curve, the red curve. That's the nitrogen exchange curve. The zoom is for 8 hours, and the big picture is all 5 days. It gets quite noisy because urea degrades, so you get a noise. But you see, and fitting this data with an exponential fitting delivers us the apparent rate constant with an acceptable precision and or error, and the black curve describes the rate of urea decay. We had to fit it with two curves because as long as a glycerol is abundant, urea degrades much faster, the fastest thing that happens, until all or most, or actually all of the inorganic phosphate is consumed, and then it degrades at the same rate as the nitrogen exchanges. Very logical. The next fastest things are, this is a summary of course, the PI consumption when glycerol was around, the PI consumption when monopalmitoyl glycerol was around, about the same values, and then you see other derivatives that develop and degrade and so on. So this was a nice thing to get the kinetics of solid liquid mixed reactions, but we also used other things than NMR. So we injected many of these reactions in HPLC, We coupled it to the mass. You see here, for example, one typical chromatogram, MPG with C13 urea and phosphate heated for 5 days. This big green broad signal here, that's the main compound, that's phosphorylated MPG. This column is specific, so it doesn't the phosphate smears, but then the other compounds are interesting, and we need mass spectrometry, high resolution mass spectrometry. So what we see, you see here a typical palette of compounds that are produced when you heat MPG with inorganic phosphate at 115 degrees. What shall I, here for example, you see glycerol, 2 glycerols on 1 phosphate. In natural phospholipids, there are the PG, the glyceryl phosphate heads synthesized by biochemistry, by enzymes, but you get them when you heat such simple compounds with inorganic phosphate. You see also carbamates and carbonates, cyclic carbonates. For ribonucleosides, one example, reverse phase HPLC. One nice thing about ribonucleosides, of course, is that you can also measure the absorbance at 260 nanometers, so we can use the extinction coefficients of the bases to quantify through UV. The colored peaks that you see here are the ion current peaks that then can be identified through their mass, and so what we get is what we get is nucleotides with phosphates all around the molecules, quite a lot of carbamate because we did this reaction in urea, and then what you see here, the most interesting, I would say, for people who connect to, for example, Holliger or many colleagues who are interested to get RNA, of course, more than 2. But the largest molecules that we could find were dinucleotides with 1 or 2 phosphate groups in the molecule. We also see adenine carbamate, guanine, inosine, hydrolysis products or depurination products, but not as much as one might think. So 115 degrees for 5 days produces maybe 10% of these artifacts, let's call them, or modified compounds, and the rest is have the mass of normal RNA molecules. So I wanted to run this because it looks a little bit, 50 peaks, 55 peaks. Anastasia, she's really tough. She just analyzed it through and quantified it through all the peaks, and that was just one experiment. And I have to say, we counted 450 experiments that were analyzed by NMR. So, you need 3 years for that. Sorry. One other example, we wanted to enhance the mixture, so we added to nucleosides also glycerol and 4 alkanoic acids, C10, C11, C12, and C13. Just so with urea, just so we see how complicated can we get? Do we get actually lipidic nucleotides like some of our colleagues could make, but not in solid mixtures? So I don't show you here a zoom into it. It's quite similar to the one without glycerol, except that you see glycerol esters on the nucleotides. What you don't see is alkanoic acids. These carboxylic acids just don't join. And that's one of the problems chemists have, prebiotic chemists have. There are not many activating molecules known that produce esters from phosphoric acid and esters from carboxylic acid at a time. There are, I would like to talk about them, but not in this talk because I would lose too much time. Then we were also interested in the volatile reaction products, so we could really pin down the mechanism of at least this urea-assisted reaction. So what you see here is a round bottom flask with 2 traps. We have argon with a very steady argon flow. We trap into the traps nucleophiles and electrophiles. Of course, in the round bottom flow, several grams of, what is this, glycerol or MPG, monopalmitoyl glycerol, an inorganic phosphate, either labeled or not, and 2A, 2B, 2C, these are ureas with different labels, and 3B and 4B, this is formamide and acetamide. So what do we do? This argon flow carries over all the gases that are evaporating. Ammonia, CO2, and water for sure. In the first trap, we have benzoyl chloride, so we catch the nucleophiles. The ammonia and the water will show up as benzamide and water. If we use N15 urea, it will show up as, it should show up as N15 benzamide. CO2 is not a nucleophile, on the contrary, it's electrophile, so it will go through. And cyanic acid, isocyanic acid, also could maybe go through, but didn't. CO2 was then captured in a cooled trap number 2 with a Grignard reaction, and so it would show up as benzoic acid. And if we had HNCO, then this would be benzamide in trap 2. If we use, or when we use C13 urea, the C13 will appear in the benzoic acid, and when we use O18 phosphate, then this O18 should actually also show up in this benzoic acid. That's the summary of the setups, the experimental setups of these reactions. And now for the results. We were interested in the mechanism. Urea is the most important condensing agent because it hangs around along, and there is no question about a urea being a prebiotic molecule. So Henri Charmet said, yes, ideal for a DFT calculation. And in the literature for decades, there are 2 stories about how urea can activate inorganic orthophosphate so that it will be able to esterify with any given alcohol or any given nucleophile, actually. And one quite old hypothesis one can describe as an associative phosphate dehydration pathway. So a bit of theoretical chemistry. What Linda then did is she listened a long time what I was telling her or them, and then she had this idea. She will put in dihydrogen phosphate, 1 molecule, into urea. She will surround it with 3 urea molecules. And now you see a blue way and a red way. The blue way is called U3WO. This means 3 ureas, no water, super dry, unrealistic in the sense that our mixtures always contain some residual water. Yes, maybe 10 or 15%. We're measuring that. It's in the paper. And the U3W2 is she added 2 water molecules. They are assisting. It's not for the hydrolysis. They just are in vicinity and maybe can change the energetics. So, and then, of course, the relative Gibbs free energies were calculated. So on the ball and stick models, you see all the molecules on both pathways in this associative mechanism. I just drew, for simplicity, 1 urea and 1 dihydrogen phosphate. What happens? Well, the oxygen of urea will can interact with a phosphorus atom such that through a transition state which is very high in energy, and it already shows this is not possible, everything higher than 40 will just never happen, will be able to expel 1 molecule and produce such a mixture between, it's a carbamimetic phosphate, yes, a mixed compound which is not very stable. It will then stabilize itself by expelling again urea in the intermediate 2. It will actually produce something that has been for a long time postulated to exist, or the metaphosphate in this cluster, yes. And then when Linda adds methanol molecule. This methanol will attack the metaphosphate and produce methyl ester and will release urea again. If we use O18, we should see O18 labeled water because it's really comes from dehydrating the phosphate. So in the associative dehydration of labeled phosphate, the production of O18 water must be able to show, and urea is not consumed because it's organocatalytic reaction. As I said, 60 kilocalories, 90 kilocalories, forget it. It's too slow. So there is another mechanism which is much more intuitive for a chemist like me. When you heat urea, you degrade it. And when you heat it in the presence of an alcohol, it will do something with it. So here you see the environment U3W0, so the super dry environment. And it contains many more intermediate states. I'll walk through certain, the most important ones. So we have dihydrogen phosphate and urea, but now in the first step, which costs more than 50 kilocalories, the urea tautomerizes into a Zwitter ionic form. The proton of the hydrogen of one nitrogen jumps on the other nitrogen, and this is catalyzed by dihydrogen phosphate. So this tautomer is needed to then deconnect the CN bond and produce one ammonia and isocyanic acid. And in the next few steps, intermediate number 4, Linda added methanol, and this methanol would then attack through transition state number 6. You see a difference of 26 kilocalories. This is quite slow. Into something which is a phosphate ester, a phosphoric acid ester, and carbamate, inorganic carbamate. That's the most stable intermediate or product. When the phosphoric acid protonates this carbamate, it will become less stable and will be able to expel ammonia. The free energy goes up. That's interesting, but of course we don't care because it's an open system, and CO2 and ammonia will just evaporate. So this will happen anyway, slowly because of TS1, but it can happen. In the last of these 4 calculations, quite heavy calculations, they take weeks, each of them. We have water which now, on the same pathway, modulates the energy. So this assisting water helps stabilizing the transition states. And so we have a similar passage into tautomerization, which costs now less than 20. That's very nice. This tautomer urea tautomer will then pass a trajectory determining transition state of 30 kilocalories, so the difference is 20 again, into something where cyanate has happened. So we cleave a bond, and then in the present, this cyanate will be attacked by the phosphate. So we see in the intermediate number 4 something similar as before, but one ammonia is already cleaved. Now when we add methanol, int 5, then there is again a transition state 6, which is quite high, 30 kilocalories in presence of 2 equivalents of water, which will then lead again to the same products in the same order of stability. So as a summary for these calculations, while the associative labeled phosphate dehydration produces water, the dissociative activation produces CO2 with 1O18 from the inorganic phosphate. And we are free to use C13 urea, and then the C13, the CO2 that we detect in trap 2 should contain a CO13 and 1O18 and 1O16. The results, the experimental results. I have to summarize strongly. I mean, if Anastasia saw that. No, she knows this table because we have it in the paper, but 450 experiments in one table, including calculations. So look first at, let's look at the calculations again. The calculation, the associative way is A, the pink background. What did we find? No O18 in the benzoic acid of the nucleophile trap number 1. What did we see in the nucleophile trap? We saw benzamide, hopefully, of course, yes, we saw it, and labeled and not, according to what we used as a starting compound, and sometimes we saw just a little bit of benzoic acid. This benzoic acid was unlabeled in the O18, so this means the residual water managed to evaporate with the ammonia and produce some benzoic acid, and this water is unlabeled. It's just the residual water that is bound in the urea crystals before they are heated to 115 degrees. In the electrophile trap, we see benzoic acid with 1 C13 and 1, indeed, O18 atom. So already this tells us the dissociative phosphate activation mechanism is operative, the association is just not possible. What we also saw was some C13 benzoic acid, which is produced from cyanate hydrolysis with water before it evaporates. And some of in the late samples, we also found benzoic acid with 2 O18 labels, which can only be explained by the fact that hot, long time reactions with this intermediate, the carbamoyl phosphate, will exchange the isotopes before it will react with any nucleophile that is present. So in a summary, what happens experimentally is inorganic carbamoyl phosphate is generated from this inorganic orthophosphate and urea through the in situ formation of cyanate. This CPI, this carbamoyl phosphate, is then either rapidly hydrolyzed or alcoholized by any alcohol that has been provided to the milieu. It produces usually the major products are phosphate esters and minor products are carbamates. The alcoholysis of CPI is actually quite slow when we use monools, monofunctional alcohols such as methanol. And this can be explained by this, already the calculation shows us 26-30 kilocalories is slow. It's possible, but slow. It would be faster if the associated mechanism would be operative, because 20-23 only for the alcohol is there, but TS1 in this pink mechanism just is not generated. What we also can say is diols and triols are much faster than monools. We did not calculate it, but we measured it in our mixtures. So the experimental products in the C line, the yellow C line, what you see is the glycerol line and the monopalmitoyl glycerol line. All these products, in the order as I drew it, are formed and partly again degraded. The carbamate cyclize, the diphosphate happened later. We have this molecule, for example, is produced the latest. Similar things happen with diol MPG, but also with nucleotides and with nucleotides in the presence of glycerol. The scope. So let's compare these alcohols that we use. Number 5, number 6, number 7, 8, 9, 10 again. What we are doing here is we show in one scheme only the maximal PI consumption under neat given phosphorylation conditions. And what we compare is cyanamide with urea, with cyclic trimethaphosphate, with the thiophosphate, with a caniphite analog, with the normal inorganic phosphate. And as you can also see struvite because it's written there. So the total conversion, when we say total conversion, is actually the PI consumption, yes? or the consumption of the inorganic phosphate material. And number 5 and number 6, first line and second line in these colored boxes, this is glycerol MPG, and 7 to 10, you hardly see any significant values. So we were actually, in the beginning, I was a little bit surprised actually by that. I mean, okay, the calculation showed that it's slow, but you never know, yeah, whether the initial state of the calculation was could have been manipulated or modified in a way that the beginning would be faster and so on. But we measured it experimentally. So dodecanol, BTGs, that's bis 3-decanol glycerol, number 7, DOG, they would just not be phosphorylated. Nucleosides are diols or triols. They were phosphorylated, and the values are quite high. So here, I just show one example. Nucleosides, a nucleoside mixture of all 4 nucleosides, ribonucleosides with urea and inorganic phosphate. And the main compounds that you see is the monophosphates, the cyclic one, of course, 37%. That's quite a lot. The rest is other compounds, as I have already shown earlier in this talk. And what I can say here as well is that we have found out under such neat dry conditions, we need a molar excess for purines. Purine nucleosides are less mobile than pyrimidines, so they are more difficult to phosphorylate. So once we started to use four equivalents of urea, everything was phosphorylated in the same quantities, pyrimidine and purines. However, cyanamide, CTMP, thiophosphate, they just didn't work. But we are interested in a mixture which contains all. So we got more and more focused on urea just by our experiments. As you see here, about 10% were dinucleotides with a different number of phosphates in these dinucleotides. We could enhance the presence of dinucleotides by adding glycerol and these alkanoic acids. And this is usually to the cost of inorganic pyrophosphate, the gray bars. With nucleotides, with nucleoside phosphorylations, you get quite a lot of PPI, but not when glycerol is present. I'm sure that this is a physical effect, that the fact that this is liquid and solid at the same time. I can show you one new experiment, one new result, not published yet, but quite fascinating. When we add valine, one equivalent, D-valine inhibits nucleoside phosphorylation, and L-valine inhibits even more. Maybe we want to discuss about it. There are many people, many colleagues who try to figure out the evolution of homochirality, and this has something to do with that, of course. Just a short run through the alcohols that are very weak. Dodecanol with cyanamide, urea, phosphate, and CTMP. These are the molecules that we found. With urea, we have less than 5% PI consumption. It just stays. With cyanamide, we have a few experiments where we had a lot of PI consumed, but in the end, the mixture contains a lot of carbamates and non-ionic, non-membranogenic compounds. It's a problem. With geraniol, you know, archaea have isoterpenoid membranes, yes? So that's why I chose this. With cyanamide, urea, and inorganic phosphate and thiophosphate, we see sometimes in this table 35% PI consumption, 23%, but it's very uncertain. The NMR peaks are so broad, I'm not sure about these numbers really. But in the mass, as you can see in this in the structures, we can find the masses of a monogeranyl phosphate, but it can be many peaks with this mass, many other peaks with higher mass, so 2 geranyls on 1 phosphate, and even carbamate, the M1 downstairs, you see 1 phosphate, 2 cyanamides that have been added actually to the geranyl. And then the last but not least, well, it is interesting, but PA, so pyruvic acid. in the experiments where we use cyanamide, urea, CTMP, PI, everything, mostly what we saw is condensed inorganic material. So with thiophosphate, we see these multiply condensed thiophosphate compounds. The organics was much more difficult to show. So we saw peaks, NMR peaks, phosphorus NMR peaks, that prove the presence of very tiny amount of phosphoenol pyruvate, even with cyanamide with CTMP, which has enough energy to produce phosphoenol cyanamide, produce these PEP. We also found glyceric acid cyclic phosphate, and so as organic chemistry, we start thinking PA, what you see here, will be phosphorylated on the acid first, but it will migrate to cyclic PAP, the cyclic phosphoenol pyruvate. This is quite a highly electrophilic carbonyl compound. We know that PA tautomerizes unfavorably, but a little bit, into PA. This is, of course, a nucleophile, so we can make CC bonds. We can add one PA to another one, it will be phosphorylate. We can add still another one, then you have 3 PA, 4 PA. Well, I'll not have to explain every step. In the end, up to 6 PA makes sense. If these cyclic phosphates would hydrolyze, you would get something like polyketides of a kind that are, to me at least, unknown in biological cells, such polyketides. Maybe I'm wrong. Up to 6 could produce such molecules, beautiful molecules. Well, what we did see in our mass spectra, that's a true mass negative ion and positive ion mass spectra, and Anastasia, who just stayed stubbornly and found masses which the masses correspond to the molecules M1, M2, and so on up to M10. M10 is a 5-mer of pyruvic acid. So with that, I can enter the final, a few slides, actually conclusion slides. What we did too, Michele Fiore did it. Urea, several of our samples that have been used to heat MPG with sodium dihydrogen phosphate and urea would, upon rehydration, form giant vesicles that we could see beautifully in the microscope. So let's get to the conclusion. Any kind of monofunctional alcohol that could have been a prebiotic precursor for its phosphorylation by an inorganic phosphate source was likely to be outcompeted by prebiotic diols and triols. Thence, monoacyl glycerols such as MPG is a diol, nucleosides, ribose nucleosides are faster than 2'-deoxyribose, and above all, glycerol itself, once present, it would just outcompete any phosphorylation of any other alcohols. One could think that carbohydrates that are even higher have more alcoholic functions, one next to the other. We did not study them. Probably they are very fast as well under such conditions. In such harsh conditions, double-chain glycerols would partly deacylate their central chain. I didn't show that in detail, but that's what we saw. Then become phosphorylated. So, for example, the BTG, the bis tetradecanoyl glycerol, would first form mono tetradecanoyl glycerol and then appear in a tiny peak as a phosphate. On the Earth's earliest hot and dry surfaces, monoalkanoyl glyceryl phosphate derivatives appear to be adequately robust, a single chain amphiphile kind, to be the most likely chemical starting line for the first bilayer forming phospholipids. When episodically wetted with salty water with such monoalkanoyl glyceryl phosphate, such as MPG phosphate, could have let vesicles emerge, as we have shown, that enclosed maybe short nucleotides and perhaps peptides inside them. Other small molecules of the isoprenoid, such as geraniol or pyruvic phosphate kind, could have been enclosed in such vesicles. spontaneously enclosed. And long-chain alkyl phosphate and carbamates, such as dodecyl phosphate, this simple alkyl phosphate, could function as co-surfactants that had an impact on the permeability, the stability, and the shape of primitive phospholipid membranes, but they would not be sufficiently abundant to form vesicles on their own. They might be important, but not alone. When vesicles would have to become more robust against divalent cations in high salinity in the sea, in the global ocean, stronger pH changes and heat denaturation maybe, those vesicles that were made uniquely out of fatty acids, which are studied very often by colleagues, would just not survive. So I have the impression, final slide, we have two theories of the origin of life on Earth. Two in the sense of location. One is located in the Hadean seafloor. The other is on the surface of subaerial landmasses and basins. So natural energy flows molded seafloors to locally sustain chemical reaction networks, and they are reminiscent of metabolism. And all colleagues of the metabolism first school, let's call it, they are there. Subaerial hot milieus gathered organic phosphates to provide robust compartments from phospholipids and heredity from nucleic acid, replication and translation. This is the other school. So I think we should think of the possibility that probably neither of these initial subaerial and seafloor systems could build up sufficient complexity to become life on their own. The subaerial locations lacked congregated long-term energy sources to push and push and push, unfailingly push forward the disequilibrium to a further level of complexity. And the seafloor was not a locum apt to amass really sufficient organic phosphates from the elimination of water out of their organic and mineral precursor molecules. The thermodynamics is just against. So the subaerial and the seafloor locations could have physically communicated with one another, and given the Hadean weather, through strong winds bearing aerosols and the intense rhythm of flood and ebb back and forth to the ocean, of water that contained sinking phospholipid protocells originating from subaerial milieus, arid conditions, and harboring maybe dense amounts of nucleotides falling, maybe peptides, maybe other small molecules. Once sunken to the ocean ground, they could fuse with seafloor compartments in these smokers, for example, or other seafloor environments, and take over their sheltered proto-metabolic reaction networks. And then the life as we know it could maybe begin. So with that, this is just an impression. It's the impression, I wrote impression because I cannot conclude all this just from the Anastasia's experiments, but I'm working in the field for, I don't know when I started, maybe 25 years ago, and that's the impression that I have after all these years and Anastasia's work. I'd like to show you my wonderful group, Michele Fiore, Anastasia, who joined me in the COVID area, as you see, Augustin, who is one of the co-workers, the others, Dimitri and Carolina, they did other things. You see Isabelle Daniel. I don't have a photo of Isabelle Daniel, who is a geologist. We got the main funding was the Volkswagen Stiftung. I profited a lot of cost actions. And so you see that my group was really hard working. I need to show another photo because the experiments were done here. Thank you for your attention. Sorry for such a long talk. But I'll still be happy to maybe answer a question if you have some.
Thank you, Professor Strazewski, for this really nice talk. See how everything fits together. It's always nice to see in systems chemistry that it's always on the interface of many chemical disciplines, inorganic, organic, solid state, etc. And now I open the floor for questions. If anyone has a question, just unmute yourself and ask. Yes, Professor Boily.
Oh, hi. Thanks, Peter. My name is Jean-François Boily.
Bonjour Jean-François Boily, je suis très enchanté.
I work with Angelo. I have the pleasure of working with Angelo, and I'd like to thank you to bring such an interesting topic to this monthly series of seminars that we have. Also, it forces us to think a bit outside the box. We don't do much organic chemistry or at all in my research area, but I see you have a large crowd from the organic chemistry section of the department. So I'm sure they're very pleased to hear about that. So you might have some questions from there. I really love the way you set this science in context and your really riveting description of the Hadean environment. And what struck to me was the word aerosol. You talked about you talked about the environmental setting of the subaerial environments of the Hadean. What do you know about the aerosol composition of those times?
Well, what we know, I never tested aerosols, but I have colleagues who did and spoke about it a lot, and I have the impression that the results got a little bit too much forgotten. The aerosols that they produced were with, if I'm correct, with peptides. I would have to look it up again. It is with peptides and amphiphiles. So when you when you create aerosols through a jet that contain organics that are amphiphilic, I think it's not so important what compounds. Much more important is how amphiphilic they are. The difference between the polar and the apolar part and the length and the head physically. And so many different compounds would produce aerosols. How long they stay in the air and how much they, how much they carry, I don't know. But I'm totally convinced that this should be experimentally tested much more systematically.
Yeah, because I was thinking there must be a lot going on up in the atmosphere, because you're talking about the fact, well, it's hot air. You emphasize, I mean, you have boundary conditions. You need to be below 100 degrees Celsius in this environment, because otherwise you don't have oceans. So your areas must be hot enough so that your reactions are water-free, right? Because you emphasize the fact that you don't have a lot of water, and you cannot have water to make this.
I see what you mean. So maybe I was too shortcut. I imagine that on the surface, reactions happen that are wetted periodically. You see, so the atmosphere and the water the atmosphere may be hot, but only on daytime. Nighttime, probably not. And so the rhythm, the very strong periodicity, has a strong impact on the transport of the molecules that were produced in dry environments. Once water gets in and the wind blows strongly, on any planet in the morning and in the evening, there are periods where the wind blows stronger. And when the tide comes up and produces waves, people who work in the aerosol field, they just I put it into the context of producing not in aerosols but transporting in aerosols. Maybe I should say that more clearly.
I see. Because I also thinking, this is my last comment/question, is that if you have you must have a lot of atmospheric dust. I mean, you have meteorites that are breaking up, and there must be a lot of surface-driven reactions there. If you expose those particles in hot air, even if it's moist, you're not going to get a lot of water vapor, or the partial pressure of water will be quite low at high temperature, say above 50, 60 degrees Celsius. And what it means, I mean, for us who we work a lot on those questions. What it means is that you're probably going to get submonolayers coverage of water on whatever dust you have up there, which means that, you know, if you have reactions taking place on dust surfaces, many of the reaction types that you're talking about might be actually feasible.
So you actually think that such reactions can happen on the dust while it's dust and not wet yet?
On hot, dry dust. Yeah.
Interesting, yeah.
Coexisting with low partial pressures of water, anything above 50 degrees Celsius could be an interesting setting.
You say something which reminds me of what is important actually is how volatile the materials are. Because when they are too volatile, they'll just blow off the dust, blow off everywhere, and will be not able to accumulate. And that's so important.
Yeah.
So for us chemists, this is a guideline. Everything that is kind of oily and not very volatile is more interesting just for the fact of not evaporating too fast.
This is interesting because those volatiles, if they have some type of hydrophilic functional groups, they can be tethered by the surfaces as well. So it's kind of like
that's very interesting.
Super interesting. Oh, yeah, well, you made my day, Peter. Super interesting topic. Thank you so much.
Thank you.
I think Professor Almqvist, you have the question.
Thanks. Yeah, thanks for a great talk. Very interesting and a bit off. I'm an organic chemist, but still more into medicinal chemistry and working on those type of compounds. But I was, of course, I had first the same type of question that Frasse now ended with the dust in the atmosphere and the surfaces created. That must be of very great importance. But I was also, but of course that adds yes complexity. But at what stage would you consider microorganisms working with these components?
At what stage in evolution?
Yeah, I mean when you have all these very interesting small organic compounds, at what stage would you then to add the complexity with microorganisms and what they can do with these compounds?
That's a fascinating question. I mean, in my mind, I am much earlier. Yes, we don't have living cells yet, and I tend to think that maybe the first microorganisms really were on the ocean floor. But they would never become microorganisms without what rains in from the subaerial surfaces. That was my message in this impression part in the end. You see, I'm not sure if such dry environments would allow for the evolution of microorganisms. I mean, microorganisms do survive in dry environments, that's true, and there are actually a lot, but they needed to evolve to that stage. And yeah, I'm not sure whether I'm answering it in a way that gives you the information, but I think to evolve really, you need to be mobile. You cannot properly evolve just totally dry in a dried out environment. No. So in another way, I would say microorganisms have to evolve somewhere where it's wet. They will be splashed out maybe by aerosols again, because this is reversible, of course. And maybe some of them will survive on dust. But very honestly, what do I know? Yeah, of course. I would imagine such microorganisms need to more or less calmly evolve over many generations into something that then will be able to survive in a totally dry dust, on totally dry dust areas.
Yeah, but also these molecules that you kind of show can be formed.
Yes.
are of course then of great importance for life to be able to start. I mean, they probably need these some of these molecules to get started. So it's the
But
kind of
you're right. I mean, cyanamide, for example, you can keep it in water for some time. It will hydrolyze. But to form cyanamide, you cannot have water. So there must have been areas. If we all, there are many chemists who use cyanamide. This is an old molecule in prebiotic field. You cannot have water for a long time until you start using it. So yeah, you were asking about which of these molecules. The slide that you're seeing now, this is a slide of another talk. Actually, all these molecules could play a role in the evolution of microorganisms, but very early.
yeah. No, you would like to start to see the formation of amino acids, of course, and this kind of more
Exactly.
strong.
Hydroxy acids, amino acids. I mean, I did not talk about the production of amino acids. I just wrote maybe peptides could also be enclosed, which means on these subaerial landmass environments that we were using as models for phosphorylating alcohols, you could also produce amino acids and peptides. But amino acids and peptides you can also produce in aqueous environments. They are less fussy about water. I mean, thermodynamics against, but if you deliver the material, you can produce peptides everywhere. But nucleotide phosphates, phospholipid molecules, no, I don't think so. Not in the ocean. So, yeah, that.
Yeah, thanks. Interesting.
Thanks for your question.
Thank you. Okay, any further questions?
I have a very small superficial question about the methodology in the beginning.
Yes.
And I saw in the tubes in the carousel, you used open tubes. And I was just wondering why they were not sealed.
Can you say again? We used often key? I don't understand.
Sorry,
Oh,
open tubes in the
Oh,
carousel.
okay, yes.
Yes.
Well, because the environment is open, but you might want to know whether we weren't afraid about oxygen. I mean, the problem is at the time there was no oxygen in the air, and now we have 20%. So we used open tubes because we wanted to have a irreversibility conditions that mimic what is happening on surfaces outside in the nature. We used closed systems to trap the gases, but we would transport them out. This is all on purpose. If you keep it completely under reversible conditions, you will not model the origin of life. I mean, the origin of complexification of molecules. Yeah, that may lead to life. It's very early. You cannot, if you close it completely, and over long times, as a geochemist, I would say you can calculate what you get because it's totally reversible. So we were irreversible on purpose. Is that answering?
Yes, thank you so much, 100%.
Thank you.
great. Thank you, Zoe. Okay. So if there are no further questions, I guess we thank you again for this, for coming here and giving us this really nice talk. And
It was a pleasure.
yeah, can
Thank you.
wrap this up. Thank you.
Thank you, Angelo. See you some other time. Bye-bye.
See you. Bye bye.
Thanks for coming.